Stop sharing for a minute so we can introduce ourselves. Just stop sharing for a minute.
Yeah.
All right, 11 o'clock, Sunday morning, back in person here. Nice to see all of you. Nice to see all of you who are joining us online for Chai and Why. So we should be on time and we've started at 11 exactly. I hope those who are watching us online, remember we can't see you, we can't interact with you. So please remember to put your questions and comments in the chat box or Q&A box on the Zoom webinar or in the chat on YouTube. And we have volunteers here who will be looking at it. So just before we start, remember that from next month, May and June, the two Chai and Whys, the first Sundays will be our summer specials, which will be in the Prithvi Cafe outside. And 1st of May, May Day, which is the first Sunday of May, we are actually going to be doing something on the world of design. And we will have various hands-on things looking at the world of design and creative things coming up next month. And we'll let you know what is happening in the rest of the summer specials as well. Now, today, of course, it's a pleasure to introduce Praachi Tiwari, soon to be doctor, who is a student at TIFR in the Department of Biological Sciences in Professor Vidita Vaidya's lab, where she is a neuroscientist. And she gets to play with psychedelic drugs, of course, in mice or rats or something, yeah, not in humans. But Praachi is someone who did a B.Tech in Allahabad in biotechnology, went to IIT Kanpur, then decided that neuroscience and the brain is what really excites her, and came to TIFR and is going to tell you about the work she does. Most of what she will talk about is related to her thesis work on psychedelic drugs and their impact on the brain and whether it could be used to treat diseases. So on that note, all over to you. Again, please remember, the way we will do it is we will see if the online audience has questions first. We'll take them. After that, we will stop the online transmission. We'll go and have chai and then we'll come back and then we can interact as long as we want. But not as long as we want till they throw us out. Let's limit. So I should also acknowledge that we have volunteers. There's Amartya there, Darshana here, and a few more people who are going to help us with questions on YouTube and Zoom. So thanks for volunteering. And over to you.
Thank you.
Yeah.
Thank you, Arnav, for that great introduction. Hello, everybody.
Screen share.
I'll just share my screen for online audience. Is my screen visible on online?
Why is your next slide also on my screen?
No. What happened? Oh, you're sharing, unfortunately, your
screen one, screen share.
Okay.
You need to share screen two.
My bad, sorry. Better?
Much better?
That's it.
All right. All right. Hello, everybody. I'm so glad to be here in person with you all today. Today, I'll be talking about some things about psychedelics that we know, some things that we don't know, the history of it, what power they have and what they can do to the brain, and how much we don't know about it. And there's so much to know. So before we go into what psychedelics can do, it has a very fascinating history, which dates back not connected anymore.
Just use your mouse. Now that works. Your work.
Laser chal raha.
Just mouse works. Just check the mouse works.
I was working. 1 second. Just stop sharing. I don't know what's going on. Should I switch it off and on again?
एक बार निकाल के लगाओ.
Your mouse, this is normal arrow key or mouse or something work to go to the next slide?
No, neither is working.
Presentation
Neither
working.
is working.
Neither
Presentation.
is working.
Okay, just give us a minute. We'll fix this little technical issue and maybe just stop sharing and
Yeah,
check it out. There's something between the presenter mode and
I think so. But it was working, Abhi,
Yeah,
so
yeah.
we tried.
right. Yep. Okay.
Yeah, working. So, okay, I'll go to the history of psychedelics first. Use of psychedelics dates back to 4000 BC, like way before, around 6000 years before now. And it all started with South America, Central Africa, Northern Europe, where there's a lot of there was a lot of culturally ingrained use of psychedelics in the names of, and they were all, most of them were plant derived, like peyote, which is a cactus, psilocybin, and ayahuasca, which is a tea root from plant. Okay, so these,
Is psilocybin also a mushroom?
Yeah.
Looks like a mushroom.
yeah, so psilocybin is actually magic mushrooms, as we popularly call them, or shrooms. And they are very widely used in psychedelic work. Yes. So how do we know they have been there for so long? 4000 BC up to 15th century AD, we have evidence in terms of cave paintings, temple murals that show usage of these drugs, these plants, especially if you can see mushrooms right here, and these plants right here, which clearly depict that culturally, traditionally, these plants were being used for years, way before we knew what they can do in terms of medical science. So what are psychedelic substances? What they do is they essentially alter your perception of the world, which is known as hallucinations. Okay. There are many different kinds of psychedelics. It's just an overview. There are way many more. Some of them are naturally derived. Some of them can be chemically synthesized. The natural ones, psilocybin, magic mushroom, ibogaine, again, it's iboga plant. Then mescaline, this comes from the peyote cactus, which is traditionally used by the Native American people. And then DMT and ayahuasca, well, clearly, if anybody has read about the South Americas and the shaman practices, they know what ayahuasca is. It's a tea, and in their ceremonies, it's a whole cleaning ritual in which they use ayahuasca for their practices. So, when did we come to know, and when did it start for?
Question.
Yes.
What about bhang?
Okay, yes. So there are many different classes of psychedelics. Okay, so the question that came online was, what about bhang, marijuana, cannabinoids? They are also a class of hallucinogens, but they're not classical psychedelics. I'll come to what classical psychedelics do in terms of what these are. Most of these, not all of them, but most of these, for example, MDMA, psilocybin, ibogaine, mescaline, DMT, LSD, all of them work on a neurotransmitter called serotonin. So serotonin, also called the happy hormone, is involved very strongly in modulating your mood in the brain, and especially in anxiety and depression-like behavior. So they all act on serotonin. Bhang or marijuana or cannabinoids have their own class of receptors, cannabinoid receptors. They work in tandem with other monoamine neurotransmitters also, but they're not classical psychedelics. So there are multiple different categories. But today I'm going to talk about this particular category, which is classical psychedelics. So how do we know? In the past 150 years around, a lot of work happened, which has led us to where we are today in terms of understanding what psychedelics can do to the brain and what kind of power they can have to attend to mental health and mental health situations. So if you see back in 1897, I'll go through it properly. In 1897, the active component of peyote, the cactus, was first synthesized. It's called mescaline, and that can now be synthesized in lab also artificially. And a lot of things happened with the advent of MDMA, which is popularly known as Molly or ecstasy. And then Albert Hofmann coming into play and actually revolutionizing the entire field with the synthesis of LSD. So these are different chemical structures. We don't really have to go into the details, but yeah, these are different chemical structures, and all of them have very similar structure to serotonin, the molecule that acts on its receptors. Now, this receptor, 5-HT stands for 5-hydroxytryptamine, which is the name for serotonin. So there's a two-way class of receptor, which is activatory in nature. GQ is activatory GBC. Okay, so all these can bind to 5-HT2A receptor, sometimes strong, way stronger than serotonin's own neurotransmitter can bind to it. Okay, and that is why it leads to a cascade of effects. And as Michael Pollan says, neuroscientists say cascade of effects when they don't exactly know what is happening. All right, so these are the different kind of structures and molecules that primarily act through this receptor. They act on other receptors also. But the hallucinations are known to be caused by this particular receptor. Okay. Now,
what is that thing you do, that little wavy, that things going up and down? Is that like a cartoon of what the receptor looks like? Or yeah, what is
this?
that? Yeah,
oh,
what is that drawing?
So, my bad.
There are two black lines and there's some red squiggly thing that.
So I have just asked, what does this cartoon mean? So I'll just briefly explain. When there is a cell, okay, it has a double layer. Lipid bilayer is what it is. So a cell has a membrane, outer membrane with two layers, on which this is a receptor. So GPCR are G protein coupled receptors. G stands for GTP. It derives from GTP because it uses GTPase for in creating that effect. So these are transmembrane proteins. They cross across the membrane, and there are 7 transmembranes. So if you can see 1, 2, 3, 4, 5, 6, 7, and these membranes are important. These transmembrane domains are important because it defines their binding pockets to the neurotransmitters, their catalytic activity. Different kind of things are defined by these different domains of the transmembranes, so that
so basically, on the cell membrane, there are regions where serotonin would come and attach.
Yes, yes.
And the same thing is where all these other molecules also come and attach.
The thing is, because of their structure, the way the binding pocket changes, its conformation, it differs for every different kind of molecule, which causes a different kind of binding affinity between the molecule and the receptor. And that is why it changes how snugly this particular drug can fit into this receptor as opposed to something else. That is how we get to know how strong the binding is of any molecule to any receptor. To go into the details of what the timeline is, because it's actually a very interesting timeline of what happened in the last 100 years, because it was all ups and downs. First, it was all enthusiasm of finding out that there are things like this that can be synthesized, naturally available, and what all they can do in the brain. It just spread like wildfire. Because it spread like wildfire, it had to be controlled. So it all started, little by little started from 1897, but it was in 1938 when Albert Hofmann was working in a Swiss laboratory to understand, I think, respiratory diseases and looking at different plant derivatives to see what can help. But he synthesized something called lysergic acid diethylamide, okay, LSD, or acid that is known in junk. Now, what happened was, for 5 years, what he was working on, it was not helpful for that. For 5 years, it was on the shelf. It was in 1943 when he accidentally ingested some of it. That is the fun and only experience with LSD, which was innocent and devoid of any expectation. What happened was, he thought he was going mad, and he took his bicycle back home because it was wartime. It was World War II time, and vehicles were not available. It was 19th April, 1943, and till date, the LSD enthusiasts, they celebrate this day as Bicycle Day. And this is one of the posters from, I think, 2040 to tell that Bicycle Day is happening and everybody is invited. So everybody just rides their bicycle the entire day on that day. This is LSD.
Ride the bicycle.
Not on LSD. No, that's illegal. You can't do that. The same person, Albert Hoffman. Do you know what else he synthesized? Anybody else knows? Okay, the same person who synthesized LSD was also the one who isolated psilocybin from the magic mushrooms 20 years later. Okay, in 1938, he made acid, and in 1958, he synthesized psilocybin from magic mushrooms. Now, this one particular person, this one scientist has given the world two main psychedelics that are being used in almost all clinical and preclinical work to understand what psychedelics can do to the mind. There are many more, but these two are being used the most, psilocybin more than anything else. Now then, people started looking at it more carefully to understand what all must be going on. There, in the background, neuroscientists started using it for understanding substance abuse disorders, for addiction, anxiety, depression, everything they started working on. But at the same time, it came on the streets. Okay, and that is 1960s and 70s when everything changed. It was a very short-lived life at that point of time for something that powerful made in laboratory that can have such kind of effect on mind. So what happened in the 60s was we came across hippie counterculture, which did not allow, which just use the word loosely. It started being used for recreational purposes. And it became a problem to control a lot of things. So because of this, most of these drugs were scheduled by UN in the US. And by scheduled, it means they're very strongly controlled, one being the most strictly controlled and 4 being least strictly controlled. And this is, I think, 2020 or 21 schedule. I'm not sure. I don't remember actually. I have it written somewhere, but I don't remember right now. So the UN status of different psychedelics. All right. Now I just want to give you a sense of what psychedelics can actually do. In spiders. Okay, so in 1995, NASA did an experiment to understand what different kind of drugs can do to spiders when they're making their web. I'm going to show only 3 components, 3 items that they used, okay? But they have used many more. So it's a very interesting study. You can take down the name of it and you can look for it. It's available online everywhere. Not on NASA website though, for some reason it's blocked there, but there are other places which have cited NASA and redirected and have allowed for PDFs to be shared. So what they do is, if you see, this is what happens to how they make the web, spiders make the web when there's no drug on board. Give some drug, LSD or peyote. Peyote is the cactus which has mescaline. They're not able to make it. Okay, it's really disrupted. But just as an aside, I wanted to show this also for everybody who likes caffeine a lot. This is what happens on caffeine. It's better on LSD and peyote, but not on caffeine. Maybe we need to think about having caffeine a lot more than. Anyway, I'll not finish that sentence. Sorry?
Our brain function performance actually enhances performance.
With these, you say?
Actually, just repeat the question.
Yeah,
was,
so the question that came was, our brain performance increases with caffeine? Yeah, well, I mean, there are a lot of things that help you, a lot of plant-derived things that help you enhance your brain function. Tea, coffee.
But here the spider is showing a completely different way.
Yes. So there are a lot of studies. I'll not go into the detail of it, but there is entire set of studies done with caffeine, caffeine addiction, caffeine withdrawal. And this is what spider is doing. I mean, things can be different in humans. We cannot really translate. Okay, but yeah, definitely. Spiders are not able to make their webs on caffeine. All right. So just to give, I don't know what will Spider-Man do. Somebody just said Spider-Man in the audience. I don't know. Maybe we'll just see all 3 generations of Spider-Man and see how much coffee they were having before making their webs. Okay. So coming back to the timeline of what psychedelics did in the last 100 years, looking at the scientific timeline, you see around 1970, the Controlled Substances Act was passed in the US. Something called War on Drugs was launched. And as of now, it seems like drugs are winning. because because it was controlled and we could bring it back in a very controlled manner, we are able to understand much more freely what these things can do. So if you see, this is just like stats of how much work has been done from 1955 to 2022 with different psychedelics, DMT, ayahuasca, psilocybin, LSD, and another new one, DOI. If you see, just have an example, look at the example of LSD. Okay, you see how much work was being done initially, and then the ban happened and it all dipped down. It did not completely end. It did not stop, but it did go down. And then it has started coming back again in the last 20 years. Okay. And if you see how many clinical trials are being done, they're just being studied more than ever in the last 2 decades. It's increasing in number. The clinical trials are increasing in number, and people are able to understand how important they are to understand neuroscience, the state of mind. So we are living in psychedelic Renaissance era. Okay. So if you see from 2017 to 2020, we're living in a very exciting time for these drugs to be studied. Because if you see right from 2017, FDA has started granting psychotherapy breakthrough status for many psychedelic assisted therapy, like MDMA, and then in August again Compass Pathways, which again it works on psilocybin to understand their role in mental state and mental health disorders. Then, of course, other places they started decriminalizing the use of psychedelics. They were not that strongly controlled, especially when it came to scientific work, especially when it came to studying neuroscience. And as you see, more and more, like I think by 2020 or 2019, and even New Zealand, Australia started their clinical trials. And as of 2021, I think now there'll be more than 300 active, soon to be active, or done clinical trials to understand what these can do in humans itself, let alone preclinical studies. So what are the different areas that we are looking at when we study psychedelics? A lot of it is related to depression and anxiety disorder. Most of it is substance use disorder. And these two things were being studied even before the bans happened. Okay, right back in 50s and 60s, people were working on this. And most of this work is being done with psilocybin. Ketamine is a dissociative anesthetic. It is not a classical psychedelic, but it can cause hallucinations. LSD, MDMA, ayahuasca, DMT, all of them also being used. Trauma, definitely. Other different mental disorders are also being studied. And how they're being studied, I'll just show you a few examples, very few examples of number of studies that are being done. So, obsessive compulsive disorder, treatment-resistant depression, tobacco addiction, major depressive disorder, patients with terminal cancer or advanced stage cancer, those have been really important studies because in terminally ill patients who are completely stuck in their head because of nearing end of their life, they often have depression and anxiety disorders. They do go through that. And these studies have shown that it helped them relieve themselves of the state of mind to a great extent. So clearly it has a lot of value. Now, what values does it have? How does it do that? In clinical settings, it is how it creates potential therapeutic effect is by emotional, social, and self-processing. In emotional processing, there are actually preclinical studies also done with octopus, where with MDMA, they've seen that social interaction increases in them. Okay, so they interact more socially. So they interact socially more than they would do without drug on. And the connectivity of these centers which modulate these kind of behaviors, that also changes. Social processing, okay, again, increased empathy and reduced rejection sensitivity, because what anecdotal stories have told us, what anecdotes have told us is that people start thinking more for themselves than about what the world will think. And then they have a clear, no doubt in their head about what their set path is. So it becomes better for them to process themselves socially.
These all clinical trials were not on humans, right? They were all on
So clinical trials are humans, preclinical trials are animals. So there's a lot of clinical studies done, and they're very, right now, actually, entire renaissance with clinical trials it all began with John Hopkins. Okay, a lot of it, there's a huge history involved in there. But these things don't happen the way we think, it's not like taking a drug and going to like a concert and just enjoying, not that way. These are very controlled way. So how it happens is you have guides in a room and they tell you how to go about the process. They tell you beforehand what to expect, what to not expect. Okay, so there are two things, the set and the setting. So the set is basically what you are expecting. Okay, what is your intention? What you think you will get out of it. And the setting is your surrounding. So everything about this is very context dependent. So that is why it's often said that when you are in a bad state of mind in the moment, this is not what you should go through, because you can have a really bad trip and it will be difficult to control. Okay, so when you're doing it in a controlled setting, you have guides, and by guides you don't mean psychiatrists or neuroscientists sitting there. By guides you mean, you have one person you can depend on. If things are going out of hand, there's this one person you completely rely on, whose presence will make you feel at ease. So these are really done in a very categorical manner, all these clinical studies. And before that, you have to go through an entire list of tests and interviews to deem fit for the study also. So you have to go through that entire process. So all this can be seen clinically, right? But clinically, we cannot see many more things, which is what is exactly going inside the brain. Okay, how the inside of the brain is changing. What is happening to neurons? What is happening to neurotransmitters? What is happening to receptors? We don't know. At least with humans, we can know about behaviors, what they're feeling. We have the power of language, which we understand, and then we can communicate. So here are the two things. In clinical settings, we cannot go inside the brain except for imaging studies, which will tell us about connectivity changing, but nothing invasive or, we cannot really know everything changing to the point of probing it to the end, just like going into it point by point and seeing where it ends. But we get that freedom with preclinical approaches. Preclinical approaches, animals cannot tell us what they're feeling, right? They cannot tell us, oh, I'm less depressed now, oh, I can see flowers. They cannot tell us all that. So there is that both the things, but both of these things can be understood way better by designing experiments like that. So in preclinical models of anxiety, depression, and PTSD, we see, by preclinical models, I mean rats, mice, rabbits, I think also, I'm not sure. But yeah, a lot of them. So what we see is neuroplasticity increases. By neuroplasticity means you make the brain more plastic. By plastic, what we mean is they are more malleable. They can change their form more easily. Okay, you bring them to a state where they are able to maybe rewire, reconnect neurons together. Different kind of connections can be formed. A lot of those things can happen. So what all behavioral effects can psychedelics have in acute or long-term changes? Okay. As I said, in brain, the activity of the brain changes. It's more chaos. Okay. So it increases the chaotic energy. Behavior-wise, I'm not going to all the behaviors, but yes, behavioral changes in rodent models, especially with depression. People have seen there is antidepressive effect. There's reduced depression-like effect. And of course, subjective psychedelic effects of the mind, that also changes. Long-term effects, what people have seen, especially with psilocybin clinical trials and long-term studies, is that psychological flexibility, cognitive flexibility, that increases, your mindfulness increases, so you're more aware, and your idea of how to perceive the world also changes a little bit. So there are different research domain criteria in which the work can be done. Self-processing systems, social processing systems, we've talked about, negative balance and positive balance systems, cognitive systems, sensory motor. So sensory motor is basically how you sense and how you respond to it, sensory motor gating. Arousal and regulatory systems like your sleep cycles, circadian rhythms, everything. People are working on very different research domains. By negative and I'm going to talk about a little bit about these two because although all of them act on the mental health state a lot, but you'll just come to know I'm talking about the balance system. The altered fear, threat, emotional avoidance, remuneration, rejection sensitivity, all associated with classical depressive and anxiety-like behavior, increased fear response, that goes down. Your empathy, approach, social interaction, that increases with these drugs. Okay, so there are many different kinds of drugs, many different structures, many different categories. There's wild mushroom, of course, a magic mushroom, but there are also frogs, smoked venom of this toad, not frog, this toad, which I don't know how people identify is hallucinogenic. Yeah, I mean, some person just went, I want to try this toad and see what happens. I don't know what happened there. Sorry? Also, it's smoked venom of this toad. Yeah. So I don't know how they decided to do that. There must be a history involved. I'm just not aware of it. And then phenylethylamines. Now, why am I showing this? Is there are a lot of different psychedelics that can be used for the same final point. But the point is that they all act in different ways. The way they go, like their milestones, their stops in between can be different to reach the final same destination because they don't all act on the same receptor, same system. Some have multitude of effects. Some can act on many different neurotransmitters and things like that. Okay. I want you to focus on this one, DOI, and for people on YouTube and this thing, can you see this? DOI. This is a substituted amphetamine, which has iodine for substitution. Okay. I'm going to talk about it because this is what I have used in my PhD to understand the effects of psychedelics on mental health. What is DOI? DOI is 2,5-dimethoxy 4-iodoamphetamine. It was synthesized by Alexander Shulgin, also popularly known as Shasha Shulgin, in late 70s or 80s. So remember, that time, all the psychedelics were banned. They were literally banned. They were scheduled. You could not use them. All the scientific work had gone underground. Okay. But there were few people, and Alexander Shulgin being one of them, who had the license to synthesize, very few chemists on the planet that time had it. He had the license to synthesize psychedelic substances if he could. So he is the one who got the world introduced to the substituted class of dimethoxy amphetamines. Okay. He's also the person who was able to synthesize mescaline from the peyote cactus that is used by the indigenous tribes in the Americas. And the interesting part is that in 1912, Merck had patented mescaline. Okay, and he found it cheaper and a much better way to synthesize it. So he got that also back into the entire list of psychedelics that could be used for work. Now, because these all were synthesized when scheduling had already happened, it became easier for scientists to access them, okay, as opposed to the other original psychedelics like LSD and psilocybin. And because it was banned from the streets, it did not go to the streets as well. You know, people were not anymore aware of what new things are coming because acid had taken over the entire generation, the entire planet in a different way. So DOI, DOM, there are many different classes of dimethoxy, substituted amphetamine that can be used. We use DOI, which is, as I said, that receptor 5-HT2A, serotonin 2A receptor. It has very high binding affinity for it. The receptor that drives the hallucinogenic effects of psychedelics. Okay, so that is why it becomes very interesting for us. It does not act on any other neurotransmitter system as we know of now. Okay, it does not. And it is very clean for that particular receptor of sex. Like serotonin also has multiple receptors, right? It's not just one receptor. But even in that system, it is very specific to two classes of receptors, and 2 is specific. So what are we doing in TIFR? This is a part of my PhD. We have looked at 3 different categories to understand what this particular drug can do in rodent models. First, we have tried to identify how it acts on anxiety. Okay, what is the dose? How long it is sustained? what's the neural circuit mediating it, and finally how the network changes. I am not going to get into the all the scientific jargon involved with it, but yes, we've been working on it, and all the people from TIFR who have been working on this are actually present in this hall today. Can you raise your hand, Darshana, Angarika, Amartya? So yeah, so we have been working together on this. This is a part of my PhD. I will just show you a few graphs, a few examples of what it looks like to see it in rodents to understand how they're responding to anxiety, especially when they're under the effect of a psychedelic. Okay. To understand that, we first need to talk about how we can score anxiety in rodents, right? I mean, they're not going to come and tell us, oh no, this is very anxiety inducing, I can't do this, right? So what we do is we play on their innate behaviors. What are their innate behaviors? What are their like core, hardcore innate behaviors? One is that they need to keep themselves safe, like any creature on the planet, right? The other is they need to forage. They need to explore new areas to find how helpful or useful that area is for them, right? So these are two conflicting things. You want to stay safe in a new place, but you still need to find out whether it's going to be useful. So we design mazes called conflict anxiety mazes because they are at conflict with your anxiety. One of them is called elevated plus maze. It's an elevated platform, as you can see, and it's a plus maze. So in this plus maze, 2 are open arms and 2 are closed arms. Okay. Now, yeah. So you see the animal moving, right? It moves either in open arms or it can choose to go in closed arms. So we score the amount of time animal is spending in open versus closed arms. Okay, we keep a tab on that. Now, the more time the animal spends in closed arms, the more safe it wants to be. Okay, so the more anxiety-like behavior it shows. The more it goes in open arms, the more it's okay with exploring new areas which might be dangerous, right? So it's keeping at bay its safety to explore new areas. So the more time it spends in open arms, the less anxious it is. So this is how we score. All right, so this is the paradigm I have used mostly in my work to understand what psychedelics are doing to them. So I'll just show you one graph. And just bear with me, I'll walk you through it. So we give animals injection of DOI, okay? And we let it cross the blood-brain barrier, let it act on the animals for a while. And then we subject them to this maze and see how they're moving around, and we record it. When we record it, we can see this is a heat map which tells us how much these animals have stayed in which area of the maze. These are open arms. The animals on DOI, okay, they tend to just move around in the open arms so much and stay in the open arms so much. And you can see the percent time they spend in open arms is almost near 200. And in closed arms, almost nil, right? So that's very exciting. And this is true for both males and females. And that's actually an important thing we all need to consider, because some drugs can have sexual dimorphism, which means that they can act differently in males and in females. So the field is more and more now asking everyone to look at both the sexes so that we know it's a population effect and not dependent on one sex. So clearly it happens the same way in both males and females. Rats, I should say. Okay. And then we have to see which area in the brain is doing it. Okay, now the brain, I'm just going to show a very cool animation of Allen Brain Institute. This is how brain looks from beginning to end. When you keep, so if you keep the brain from front to back and start cutting it in sections called coronal sections, which are basically going up to down from front to back, like this, you chop it up, like you're slicing, right? So when you do that, this is how it looks like. I will just tell you the different areas. So you start with orbitofrontal cortex. This is prefrontal cortex, processes a lot of cognition. It's going, other subcortical structures are also coming in between. Here is the hippocampus. You can see forming it. Then there was amygdala. I'll play it again. It's very fast because it's a YouTube video. Okay, you can see it on YouTube. It's openly available on Allen Brain Institute. You see amygdala starts forming here, which looks like an almond. This is hippocampus. So these are the areas. Why am I pointing at these areas? Because although there are a lot of different areas involved in anxiety network, these three play quite a major role in modulating anxiety-like effect in animals. So I targeted these different regions in the brain to see if I directly give DOI there, what happens. What I see is not in amygdala. This is amygdala. You see, it's almond-like at the bottom of the
Can you
brain.
just repeat it in a way that the online audience knows
Yes.
because they can't see where you're pointing?
Yeah.
So
If you
that-
see this, this is amygdala.
nothing-
I'll show it with a pointer here only. Can you see the pointer now? Yeah. So this is amygdala. You see the red ones at the bottom. This looks like almond. And this is where I surgically implanted cannulae to see, to then acutely give psychedelics when I have to subject them to the maze. This is prefrontal cortex. There are different regions, subdivisions of prefrontal cortex, the cortical region that is also involved in cognition and processing of emotions. Same as with hippocampus. Now, hippocampus is a very interesting structure because the entire length of hippocampus looks very different. If you see here, it's not playing.
You have to go back.
Yeah. If you see here, when hippocampus comes, it is a very 3D structure. Okay, it starts from now. See here, it's going. Then it keeps increasing in size in a manner of speaking and goes till the end of the brain, almost at the end. Now, why it is interesting is because what the front part or the dorsal part of hippocampus can do is very different from what part at the caudal end, at the back end, or the lowermost part of hippocampus can do. And when I subjected these animals to psychedelics in different regions of the brain, what I see is it was only hippocampus, the ventral hippocampus, this part, almost at the end of your cerebellum. This one is involved in modulating the anxiety-like effect in animals. And you see, not in these, but only in this, if you give the amount of time spent in open arms as compared to closed arms. They just tend to stay in open arms. Okay. There is a lot more work that has been done. I'm just showing it to give you a sense of how we do the work in preclinical settings. So just to summarize the part that we have done in TIFR, psychedelics, they can act on anxiety-like behavior, right? And they often do it, they tend to do it via the hippocampus, at least in my work, that's what I see with anxiety-like behavior. Possibly through amygdala, but the jury's still out on that. And the neurons, which are involved in doing that. So my work just went on to see what kind of neurons and what it does to the network and how it changes in the brain. Because that's important to understand what it's doing to the brain. Is it shutting down? Is it activating? How the brain is talking to each other, the different regions of the brain. This gives us a sense of how we can design drugs, how we can understand what are these things actually doing in the brain at different levels. Because one thing is, what I saw is that your hallucinations are processed different from your anxiety modality. Okay, so that becomes very important because clearly when you're taking systemically, when you're taking orally, when you're taking through injection, these drugs, they act upon the entire brain. So everything is happening at once. But clearly they are mutually exclusive, probably. I mean, at least with this, in this case, they are. That's what I see. So in anxiety-like behavior, what is happening is hippocampus is coming in. And hippocampus is a very interesting structure, I said, because shutting down of this this region is known to be involved in reduced anxiety-like behavior. Okay. And what in my work I see is that actually the neurons that are coming into play are the inhibitory neurons. What inhibitory neurons do is that they inhibit the activity of other neurons, like neurotransmitter. So basically what they're doing is they're shutting down the hippocampus. Okay. And the receptors on these neurons is what we think is driving the anxiolytic effects of DOI. We're trying to, again, use a few more other lab synthesized psychedelics, which are available for research work, to confirm whether that is true across different psychedelics. But this is only one part of what we are doing in TIFR, one part of science, what we can do in TIFR. Okay, there are many different areas preclinically that we can prove. We can look at it at genetic level, at genes level, what genes are changing, how immediate the responses are, proteins, what are the levels of transporter levels, and IP3, all those things. Cellular, how the plastic, how brain is becoming plastic, how they're changing their architecture, their morphology, how circuits are changing, whether they're increasing their connectivity with each other, whether they're decreasing, which becomes interesting because default mode network, at least in clinical studies, have been shown with psilocybin that if you see a default mode network, which is basically connecting your cortex to the hippocampus and other subcortical regions which are more primitive, what happens, what default mode network is, it's active when you are not active, right? So it's involving remuneration, your worries, your thought process. It's different from your attention network. So that seems to shut down when psilocybin is on board. Okay, so this is what people think is giving a sense of what you call ego dissolution or ego death, that keeps you away from yourself, because now and creates a chaos enough to break you out of that remunerative process. And that is how it can help with addiction, because addiction is something that addiction, anxiety, depression, all of these disorders, what they do is they make you get stuck in a certain loop, right? So the idea is that this chaos in the brain might help them to break out of that loop. Okay, so and then different paradigms also involved in physiology. So there's a lot to be done, and there's very little that we know, and it's a very exciting time for psychedelics, because the more we're getting to know about it, the more we know how powerful they are and how much work needs to be done to control their power in certain domains so that it just does not blast off all over again. This is the lab. This is our lab. This is my advisor, Vikita Vaidya, who has given me a lot of creative freedom in my work. And all those who could not see, Angarika is not here in the picture, but Amartya and Dashna, they have been working with me for a long time on this project. I would take questions now. I'll go back to the summary slide.
Okay, so the way we'll do this is I'm going to quickly see if there are any questions online.
Yes.
And once we are done with that, we will go out for caffeinated beverages. Now I'm going to really worry about it, but I'm not spinning a web, so it's okay. Let me just quickly see. I'm on this side just so that the mic catches me and you don't have to repeat the question. There are a few questions on plants like datura, which also apparently have been used to tweak neural connections, etc. Does that work? Is it a similar type of effect with alkaloids like scopolamine and things like that? Yeah.
So yeah, people have been working with scopolamine, datura, and the likes as well. Again, they don't come under the exact category of classical psychedelics. They're different. They act a little differently, as Arnab said, they're alkaloids. But yes, they do cause hallucinations, and their effect is also being seen in mental health state. And the more we can work on it, the better. I think in TIFR also, one of our colleagues is working with alkaloids to understand its effects on the brain. So yeah, they can, but that's the working hypothesis that they might.
Okay, and these drugs like LSD and all, do they make new neural connections? Can synapses be made by these drugs? If yes, why?
Yeah, so why? A cascade of effects happen. Okay. So it changes the essence of the brain. Behaviorally, we know a lot of things. Molecularly, cellularly, we are still trying to understand. We do know it creates more synapses. probably because it increases the firing activity in the brain. That is one of the hypotheses of hallucinations, that what it is doing is just buzzing up the brain. Now, what you do is, if you fire everything at once, okay, you might be able to break older connections and allow them in a sense to make newer ones, because now they are more active, they can reach out more to their neighbors, to put it lightly. So probably that's why they can.
Okay, I do not see any more questions here. There's one on YouTube. It says, what about opium?
Opiates. Okay, opiate crisis is a rabbit hole I will not go into, because opiates are addictive. Believe it or not, psychedelics, classical psychedelics are not. Okay, that is why they have more therapeutic potential. Opioid crisis in the 50s, 60s, 70s, that thing just destroyed an entire generation. It can do a lot of things, but it is very addictive. And the idea of psychedelics is to actually use them against addiction as well. So yeah.
Okay, what kind of lab techniques did your group usually use in the lab?
A lot of them, whatever is needed to answer the question. It starts from behaviors. We are classically a neuropharmacology lab. That means in neuroscience, we use pharmacology to understand. Okay. So what we do is we look at behavior in animals. We look at how these behaviors might, what is happening molecularly, cellularly. So we look at proteins, we look at RNA, DNA level changes. We look at first behavior. We look at circuit level changes also with respect to electrophysiological readings. We look at neurometabolism, what it is doing to ATP, and mitochondrial DNA. There are many things we are doing actually, and we use all the techniques that would be needed. That is one good thing, that we collaborate. If you want to ask a question and we're interested enough in it, we'll find a way to answer it.
Can I ask you a follow-up question?
Yes, yes.
I mean, you said you take something and go to the
Yeah.
hippocampus and put some. I mean, the mouse or rat doesn't mind, yeah? Like brain ke andar kisi ne kuch dala and now you should go run on this maze or something like.
Yeah. So how it is done? Yeah, I wanted to skip that, but thank you for asking that question. So what we do is it's called stereotactic surgery. So stereotax basically is it's a platform which has 3 degrees of freedom. So usually those kind of setups are used for neurosurgeries. Of course, they are used in human neurosurgeries also, but that's a very different kind of setup and a very different scale. In rats or mice, there's a smaller one in which you can set the animal such that their head does not move. They're under general anesthesia, making sure there is no pain involved. They're giving painkillers. And what you do is, there is a brain atlas actually available. Okay, using that, you can know which age of animal will have what size of brain. And when you're doing surgeries, there are sutures. So in our head, if you've ever seen a human skull, there are joints, right? That give kind of indentations and lines on the head. So similar things are available. Similarly, it's there in rodents also. So there is a suture which just goes like there, and there's this point which you can mark your origin, and from there you know where you are in the brain.
You can stop sharing it. You can see better.
Okay.
Otherwise, you're a very small family.
Yeah. Okay. So yeah, so that's what happens. So using that location, we will know where we are in the brain. And once we know where we are in the brain, we can put our implants again through that entire system, that stereotactic system, which gives us 3 degrees of freedom, which is front and back, anterior, posterior, left and right, mediolateral, and up and down, dorsoventral. So we have 3 degrees of freedom to know where we are in the brain. Okay. And using that, we can just, so how I'm delivering is, there are sterilized needles called cannulae, which can fix in the head when the animal is anesthetized and under surgeries. You can just fix it there, and then the animal goes through a recovery period for a couple of weeks with all painkillers, antibiotics taken care of. And after that, when it has recovered completely, before subjecting them to the maze, you can put the drug directly in the brain and then let them explore the arena. That's how we do it.
Okay. One more question which seems relevant. maybe you know this, CBD has been separated from marijuana so as to prevent the side effects of marijuana, the dependence on this compound. But why is even taking CBD, you can get addicted to it?
Cannabinoids, I mean, they mean cannabinoids, I think. Cannabinoids, I do, sorry?
Like if CBD is separated, there's another thing. If CBD is separate from the dependency of marijuana, why do you still get addicted to CBD?
If there is no, so CBD, I think, is then active. I'm not sure about that, but if there is no dependency, there should not be any addiction as well.
Okay, we'll take this discussion later. Another question which has now come multiple times: are Bachelor of Pharmacy students suitable for research in your lab?
I think so. B Pharma students can apply for.
The answer is yes.
Yeah, but anybody who's let me
Yeah,
just
if
put it out
an
there.
engineer could do it.
Yeah, let me just put it out there. Taking these drugs is illegal. We only use it on animals for research work. So, yeah.
Okay, I think there are no more questions on Zoom, and I don't see any new questions on YouTube either. So on that note, let us thank Praachi for taking us into this. And I'm going to end the Zoom meeting right now.